Hi, my name is Annaka Saffron. I'm a rising junior at Johns Hopkins University, and I had the amazing opportunity to participate in the NIH SenNet CUSP internship program in Dr. Cristiano's lab at Columbia University. I'll be talking about Opal Multiplex for senescent markers. For the background, senescence is changes in cells such as a pro-inflammatory senescence-associated secretory phenotype, nuclear abnormalities such as DNA damage and modifications to the nuclear envelope, mitochondrial dysfunction, and lysosome dysfunction as well. And senescent cell populations increase with cell replications and environmental cause of DNA damage, such as UV radiation or infrared radiation. And this causes accumulation of senescent cells in tissues as a person ages. And that can eventually lead to health issues. And senescent cells cannot be accurately characterized by just one marker. So to study them, we have to use many, including beta-galactosidase, lemon B1, caveolin 1, HMGB1, PH2AX, 53BP1, P16, and P21. And the ones that are highlighted are the ones that we use in our study. The figure on the right shows some of the effects of the SASP or senescent-associated secretory phenotype on neighboring cells and highlights the need for spatial mapping of senescent cells and their biomarkers, especially over the course of a person's life. This is where my project comes in. Okay, so Opal is a kind of spatial proteomics where a primary antibody binds to a target of interest, and a secondary antibody that's conjugated to horseradish peroxidase binds to that primary antibody. Then the Opal fluorophore is added, it gets activated by the horseradish peroxidase and stays in the area near the target or the protein that we're trying to look at, even after heat treatment removes both antibodies. Those steps can then be repeated with new antibodies and a different Opal fluorophore. The tissue now has 8 stain proteins that can then be imaged. This mechanism is illustrated in a figure from another group that uses Opal. My group is using it to view 7 senescence markers and some lineage markers simultaneously in slices of skin. B, C, and D, like in the bottom of this image, lead into why optimizing it is necessary. It shows that some fluorophores are brighter than others, and there's also epitope shielding, so the order of Opal and antibody matters. So for our methods, this is our workflow for optimizing it. So we optimize Opal staining on monoplex, which means like 1 antibody and 1 Opal per slide. And so we changed buffers in their pH 6 versus pH 9. We changed the antibody's dilution factor, the antigen retrieval time, and antibody incubation length. And then based on the results from the monoplex experiment, we decide the order of the antibodies and Opal pairings for the multiplex staining. And these results are in table 1 and 2 seen here. And then we prepared the slides we're going to use for the multiplex with control skin constructs, extrinsic senescent skin constructs, and intrinsic skin constructs, all on one combined slide. And then we performed the multiplex assay on 3 slides with extrinsic markers and 3 slides with the intrinsic markers. That's a total of 6 slides plus 1 autofluorescent slide that we sent to the core for imaging. So for our skin constructs, they're very much like regular skin. They have like the 3 layers of skin, so the adipose, the dermis, and the epidermis. And they contain cell types that are melanocytes, fibroblasts, keratinocytes, endothelial cells, and stromal vascular fraction, which is like adipose cells and a combination of a few other cell types. These are all isolated from neonatal and adult donors. The cell types, once isolated, are cultured separately until we have low passage cells for all 5 cell types and high passage, which usually means that they divide very slowly and they have cell morphology. For example, like fibroblasts will start to look like very stretched out, large, and have like kind of blebs coming off of them. For the 3 cell types, keratinocytes, melanocytes, and fibroblasts. And then we made 3 groups, control, which is just low passage skin, made from all low passage cell types. Extrinsic senescence, which is a skin construct made out of all low passage cell types, but just like the control, but then receives UV treatments for about 1 week. And then the last group is intrinsic or applicative senescence, which is a mix of high and low passage cells, but no UV treatment. For our analysis, when we sent them to the core used the phenocycler to scan regions of interest. So it looks at our skin construct slices and measures the intensity from the opals coming from those skin constructs. And then we feed those images into an algorithm in a Koya inform, which then demultiplexes the Opal signals, and we get single channel images. And then we can use those to feed into Cell Profiler to measure the intensity of all the Opal signals in each cell. And it recognizes cells via DAPI, the nuclear stain. And then we compare intensities, the biomarker expressions between control intrinsic senescence, extrinsic senescence models, and between adult and neonatal derived skin construct slices. Okay, onto our results. This is an example of our control slides and how we chose our conditions from our control slides. So the brightest fluorescence, seen as the green dots in these images, is for the 800x dilution with the AR9 buffer. It's the one that's enclosed in the red box. And so then we use those conditions in the multiplex because it gave the best image here. And then these are the slides with the skin construct slices on them before demultiplexing. So seeing the legend for the two images, some Opal dyes get their own laser channel, such as Opal 480 and 780. So 780 like only corresponds to K14, no other proteins, and each corresponding to one marker per slide, but CY, MSI, and FITC each correspond to two or more markers, and thus had to be demultiplexed so that we can use the channels. And these are the images after demultiplexing for the intrinsic senescent markers. On to our conclusions. So we succeeded in optimizing the Opal multiplex. But quantitative data collection for the experiment using the optimized protocol is in progress. And we can use this experiment to confirm the mechanisms and biomarkers of intrinsic and extrinsic senescence in 3D skin. And then in the future, our method could be used to further characterize senescent cells, secretory phenotype, and their role in any tissue. Okay, and I'd like to give acknowledgements and thanks to the SenNet CUSP program, the NIH, Columbia University Medical Center, Dr. Cristiano, Dr. Papalardo, Kavya Batra, and everyone in the Cristiano Lab, Department of Dermatology, and the Human Immune Monitoring Core. I had an amazing time this summer, and I learned so much about cell cultures and different softwares. It was so interesting, and I'll definitely use this in my career in the future. Thank you for the opportunity.